The last part in our series on muscle tissue. And so we'll begin to look at what's going on within the muscle as we have contractions occurring now that we've talked about the actual sliding filament theory and how that is going to occur. So one of the things that we have helping us out in muscles as well is that the whole muscle with skeletal muscle does not all contract at once. And so we have what we call motor units. And so these motor units are individual neurons and all of the muscle cells that it actually innervates. And so we can see here with the purple neuron here, it goes out and it innervates this one, and it also innervates this one. The green one goes out and innervates this cell, and this cell. And so not all motor units are of the same number of cells. Not all motor units are the same type of fiber either. And so we can see that we have two different kind of colors of muscle fibers there. And so they may be of different kinds in terms of the fiber types themselves that are there. But each neuron interacts with only the same type of muscle cells that are there. In so doing, we can use either, in a sense, this portion of the muscle, or we can use this portion of the muscle, or we can use all of the muscle fibers in there. And so this is how we're going to decrease the amount of fatigue that a muscle might experience by simply changing the amount of the actual muscle that contracts at any given time. This also allows us to limit the amount or the force of contraction that is occurring as well. It's not an all or nothing process. And so when we look at twitch contractions in the next couple slides here, we're essentially looking at things that happen with a experimental model. And so with stimulation of the muscle right here, and so we apply an electrical stimulus to the muscle then starts to go through a contraction. So as it goes through that contraction, we see a period of time where it does not change its force. And then this is essentially the period of time where calcium is leaking out of the cells and getting to the troponin, opening up the tropomyosin, and then we get full contraction of the muscle potentially, and then relaxation. So calcium is coming out of the sarcoplasmic reticulum. It's then being pumped back in, and we get less contraction as relaxation then occurs. This whole process can be a couple of milliseconds long. It can be hundreds of milliseconds long, depending upon the particular muscle fiber that is being stimulated that's there. So when we have a look at different types of muscles themselves, and so in the bottom picture here, we can see that we have extraocular eye muscles, we have muscles from the leg, the gastrocnemius and the soleus, and each one of them has a slightly different contraction. And so the extraocular eye muscles have a very fast twitch. Their latent period is basically nothing. Their period of contraction itself is very quick, and the relaxation is very quick. And so if you think about it, when we look with our eyes are kind of always darting around, looking at little things for just a moment, and then moving on to the next thing. Versus, say, the gastrocnemius here and the soleus, both of those, even though they are actually connected to one another via the tendon, they both also have different functions as far as what they do. And so gastrocnemius and soleus have different twitch times also. And so soleus is going to take a little bit longer to contract than that of gastrocnemius. And so when we look at those different components as far as what's going on within the latent period, the contraction period, and the relaxation period, we can see that there's some processes that have to occur. So that latent period is kind of like taking the slack out of a rope. If you're going to pull on a rope and it was just kind of piled up there, before you can actually form any tension in it, you've got to take that slack out of the muscle itself or out of the rope. In a normal muscle that's in a sense living and not in a experiment, that muscle would always have some tone in it. And so we basically have very little latent period there. The contraction period then is going to be dependent upon the type of muscle, anything from relatively fast and even lower than 10 milliseconds to upwards of a couple of hundred milliseconds as that occurs. And then essentially however long it took to contract, we're going to have that same in terms of the relaxation that's going on there. And so during relaxation, calcium is being pumped back into the sarcoplasmic reticulum. So as less and less is available in the sarcoplasm, we then have more relaxation of the muscle itself. In skeletal muscle especially, we have a refractory period. And so this refractory period we see as essentially a period of time where even if we attempted to have another stimulus, we wouldn't be able to get another stimulus to occur. It's lost its excitability. And so no matter how much voltage we apply to it, no matter how much stimulus we apply to it, it does not have that ability to contract once again. And again, depending upon the muscle, that's going to dictate how long that refractory period occurs. We can start to see the refractory period when we are stimulating the muscle at a period of time that is outside of the refractory period. And so we can continue to have stimulation occur to the muscle, and we can start to get what we call wave summation. And so if we apply a second stimulus before the muscle has completely relaxed, we're going to get a stronger contraction the next time. If we continue to do this over and over and over, our contraction force is going to increase all the way up to the maximum force that that muscle can do. If we apply that stimulus very quickly, and so we have a very quick stimulus on there, we're not going to see any kind of bouncing of the muscle. Basically, the muscle is going to go straight into full maximum contraction, and then it's going to stay there as long as that stimulus continues to be applied to the muscle itself. So this gets us either complete or unfused tetanus. With unfused tetanus, there's a little bit of a relaxation. So we see that little bit of a dip there, and then a dip, and then a dip in the muscle's force that's being generated. And so this is kind of going to see the muscle kind of bouncing or twitching as you were watching it. With fused tetanus, it just goes, essentially it's all or nothing almost. And so it goes all the way up to full contraction, stays there, and the muscle is basically going to be kind of silent. There's no appearance of any kind of relaxation whatsoever. So what this tells us is that there's always a little bit of extra calcium still in that sarcoplasm. So during that period of time of either relaxation or with fused tetanus where there's really not any kind of relaxation whatsoever, enough of that calcium has remained to where we have a stronger contraction the next time. And so essentially the muscle itself is not allowed to go into any kind of relaxation. Or with summation and unfused tetanus, we see that there's a little bit of relaxation, and that's it. And so the muscle is going to be kind of sitting there twitching, bouncing up and down a little bit as that occurs with unfused tetanus. And so it's as if, in terms of that calcium, if we had, say, 100 molecules of calcium released into the cell, and then relaxation starts and we get like 50 molecules out, and then we add another 100, so now we're up to 150, and then we take another 50 out, we're down to 100, we add another 100, and we're up to 200. And so we're constantly going up and down in terms of the amount of calcium that's inside the cell, and that's going to equate to the amount of contraction that we see as well. Within the cells, we have different motor units. And so within the muscles themselves, we're going to see that those motor units allow us to use some of the muscle and then allow the rest of the muscle to relax. So some parts of the muscle are firing, they're contracting, some of them are just relaxing. And that allows them to delay fatigue. Some of them get to rest, some of them get to contract, as long as we're not using the muscle at its maximum capacity on there. What this also equates to is that when we do contract the muscle, it's not like the muscle is, in a sense, all or nothing. And so contraction is kind of almost like a robot or something in terms of it's all on or all off. Rather than having that jerky contraction, we start to see, in terms of the contraction, a smoothing out of the contraction itself. And so it's a nice wave rather than being spikes that are there. Depending upon the size of the motor unit is going to dictate the ability of that muscle to generate a fine motor control. And so small motor units, where we have a neuron that goes out and it only communicates in a sense with a small number of muscle fibers versus a large motor unit where it communicates with a large number of muscle fibers, we're going to see a difference in the type of movement that we get as well. Large motor unit, kind of big, bulky movement. Small motor units, very fine control. And so think of like small motor units as being those of the extraocular eye muscles, or even the muscles that are controlling your fingers. And so we can make very, very small incremental movements with our fingers versus with our shoulder. And so one of the ways to kind of see this is to write with just your hand, so just your fingers writing your name, and then do the same thing, but lock your fingers in place, lock your wrist in place, lock your elbow in place, and write with your shoulder, and see the difference in your ability to generate your signature. And you'll typically see a fairly big difference between the two. Depending upon how much stimulus we give a muscle, dictates whether we see contraction or not, and also dictates how much force of contraction is going to be there. And so we're going to see a couple of different parameters. And so we have threshold, the minimum amount of stimulus that we get to where we see contraction of the muscle. Once we go beyond that threshold, now we start to see contractions. And so we start to see force being generated in the muscle itself. And depending upon how much of that electrical activity we give to the muscle determines how much force we can generate. And so as we kind of turn up the stimulus and then turn up the stimulus, we get more and more contraction along the way. And this is where we're basically adding more motor units to the contraction that is occurring. And so here we have our sub-threshold stimuli. The stimulus isn't enough to generate any kind of force, so we see nothing over here in terms of the force being generated. But once we hit threshold, we now see a contraction. We turn up the stimulus a little bit more, and even still, we see the same contraction. We get to a next level, and we see more force of contraction going on up. We turn that force of contraction up a little in terms of the stimulus a little bit more, and we see the voltage go up and up until we hit to a point where no matter how much we turn up the voltage, the contraction stays the same. We've reached the maximum that that muscle can do as far as the force of contraction goes. We would have to train that muscle to become stronger in order for it to make any kind of extra amount of force. So within muscle, typically, when we think of muscle inside the body, the muscles themselves are not either contracted or relaxed. There's not that situation where essentially everything is either on or everything is either off. We have a tone within muscles that allows for the muscle to essentially kind of stay at the ready. It doesn't create any kind of movement, so there's no movement within the muscles themselves. It just keeps the muscles essentially ready for contraction. It's going to reduce that amount of time that we have for kind of taking the slack out of the muscle. But we also use this for posture. And so within skeletal muscle, we use this for our postural types of muscles. So as you sit there, your head doesn't just fall over and fall down like a baby's would, because there's a large amount of tone there. The muscles are contracting all the time. And so as your head kind of moves a little bit more forward, it pulls it a little bit back, and then it moves a little bit forward until we pull it back. And so that posture process requires us to have some contraction within the muscles themselves. In other muscles, like smooth muscle, we're going to use this to maintain the tone within blood vessels. We're going to use it to maintain the tone within the digestive tract. And so this same process of tone is used not just in skeletal muscle, but also in smooth muscle. So when we think of muscle contraction and we think of the process of contraction itself, we typically think of concentric contractions and eccentric contractions. And so we're moving a load, we're having a process where we're moving something along. And so this is under the general isotonic contraction. And so we have concentric contractions where the muscle shortens as we generate force. And then we have eccentric contractions where the muscle lengthens. And so just like in moving the book there in the example, as we lift the book up, we generate force and we cause the muscle to shorten. But at the same time, if we want to put that book down, unless we just turn everything off, the book is going to fall. But if we contract that muscle as we allow it to lengthen, we get an eccentric type of a contraction, and the muscle has the ability to lengthen at the same time. And this happens all the time as we move around, not just lifting books. Isometric contractions are a contraction of the muscle without any movement. So this would be like our postural types of contractions, stabilizing a joint. Even in this particular situation here, the wrist in an eccentric and concentric contraction, if we maintain the position of the wrist the whole time, the wrist muscles are going to be doing isometric contractions, while biceps brachii, brachialis are doing either a concentric or an eccentric contraction. So different joints can be doing different things at the same time. So an isotonic type of a contraction, we have shortening of the muscle, so we have a concentric contraction. We're lifting the weight, we're able to move that weight, and we see that we generate force, and the muscle itself is shortening. Versus an isometric contraction, we now have a weight here that the muscle is unable to contract, and it's generating force. So as we measure the amount of force that it's generating just under 6 kilograms. So the length of the muscle stays the same. Now, one way that we could possibly get that muscle to contract and shorten would be to change the length from the beginning. And so to alter the length of the muscle from the beginning point so that the length of the muscle is starting out down more in its optimum. And then perhaps it's going to be able to generate a little bit more force before it starts to fail. So just like we talked about the length of the muscle having to do with the force of contraction that can be generated. Not all skeletal muscle fibers are the same. So we have different kinds of muscle fibers. We have red muscle fibers and we have white muscle fibers. And then we can also talk about how quickly the muscles can actually fire off within them, so the speed of contraction as well. So red muscle fibers are kind of your darker colored fibers. They're going to have essentially fatigue resistance because they have more myoglobin, they have more mitochondria, and they have more capillaries supplying them with everything that they need. Versus white muscle fibers have less of all. And so they have less myoglobin, they have less mitochondria, less capillaries, so they're going to be less fatigue resistant. They're going to be meant to work and then relax, versus red muscle fibers are going to be able to work in a sense all day long. The speed at which they can actually contract is dictated by the ability to break down ATP. So we said that we had to hydrolyze ATP and take that ATP and break it down. So this is done with an enzyme, so ATPase, that's there. And we're going to hydrolyze ATP into our ADP and inorganic phosphate. And so we're going to get the phosphate group coming off of there. And the quicker that we can do that, the faster that muscle can contract. And so we can talk about the twitch of a muscle. So in the picture there, we see that a muscle here has 3 different kinds of fiber types. And within any given muscle, we're typically going to have a blend of the different types there. But in this particular one, fast oxidative seems to be the majority of all of the muscle fiber types. It does have a little bit of slow oxidative, and it does have a little bit of fast glycolytic within those. And so again, the difference determines how quickly of a twitch they can make, and also the fatigue resistance. And so slow oxidative, our slow twitch fibers, these are meant to essentially work all day long. And so this is going to be maintaining posture types of things. And so long, prolonged contractions, relatively slow twitch for that. From there we have fast glycolytic. And so these fast oxidative glycolytic types of fibers that we have the ability to contract and contract and contract and contract. They can go relatively quick, so we can do a quick sprint if we need to. But overall, they're going to be kind of used for things like walking, kind of everyday, all day long types of movements. And so things that we can continue to do over and over and over. Versus fast glycolytic, these are going to be our power muscles. And so these are our muscles that they're meant to contract and then relax. And so this is things like weightlifting, where we want those muscle fibers to be able to contract very powerfully, but then they get to relax for a while. So we have a combination of muscle fibers within a given muscle itself, but we do see kind of specific areas that are going to have different types that are there, depending upon kind of a more proportion, a higher proportion of a particular type of muscle fiber that's there. And so things like our postural types of muscles are going to have those slow oxidative fibers. Those muscles that are meant to do long, sustained, or fast contractions, lots of power, and not really do a whole lot, are going to be our glycolytic fibers. Think of like raising your hand to ask a question. Your shoulder isn't going to last forever in terms of holding your arm up, because they're using those fast glycolytic types of fibers. If a muscle fiber type is being innervated, it's going to be innervated by the exact same neuron. And so one motor unit has the same types of fibers. So they're either all fast glycolytic, they're either all slow oxidative, or they're all fast oxidative fibers, depending upon the type that is there. So we can't cross between one neuron supplying each different type of muscle. When muscles grow, we need to add proteins to those cells, and one of the things that can occur with that is normal fatigue of the muscle. It tells the muscle that it needs to increase in its muscle fibers, increase its strength. But we can also kind of circumvent this process by using things like anabolic steroids. And so performance enhancing types of drugs can work at the muscular level itself to increase muscle size, strength, and endurance. The downside is that it doesn't just affect the muscles. And so it's not only affecting the muscles of the skeleton, but it's also the liver, in a sense, has to deal with those excess steroid hormones. The kidney has to process them as well. The heart becomes affected by those increased numbers, and all of these processes can be very lifelong. So even though a person may use them for a short period of time at times, if they overused them and caused any kind of damage to the other organs, that's going to be long term. It's going to stay with them essentially for the rest of their lives. Moving on to cardiac muscle for a little bit. You'll talk about cardiac muscle in the heart chapter itself, but we'll look at kind of the physiology here of individual cells. So we talked about them before. They are short muscles. They branch. So we have branches in them. They have a single nucleus. We have our intercalated discs with our gap junctions to provide for communication from one cell to the next that is there. And when you think about what a cardiac muscle has to do, having all these branches and short cells makes sense. We wouldn't want big, long cells within the relatively small heart that is there. Since cardiac muscle has to work, in a sense, all day long, they have a large number of mitochondria. The space inside the cells is relatively large, so there's a lot of fluid inside, a lot of sarcoplasm that is there. So it has some things that are more, it also has some things that are less. And so a less well-developed sarcoplasmic reticulum means that calcium isn't stored in large amounts, which means that the calcium has to enter the cell from the extracellular fluid. And so a large amount of that calcium is actually going to come from the outside of the cell. And what this equates to is a contraction that lasts longer than that of skeletal muscle. And so whereas skeletal muscle can be down in the 30 millisecond range, cardiac muscle's up in the 300 millisecond range. And so it has a longer contraction when it's relatively at rest. So the T tubules are less prevalent in that of cardiac muscle. The sarcoplasmic reticulum is less in cardiac muscle, and so that means that more of it is going to have to come in terms of calcium from outside of the cell. Cardiac muscle does have the benefit of having autorhythmic cells, so it does have the ability to set its own pace, and that pace is also dictated by the nervous system. And so the autonomic nervous system is going to bring down the natural pace of the heart down to about 75 times a minute or so. And that's going to require huge amounts of oxygen. Lots of oxygen, lots of glucose coming into the muscle cells. And so just imagine doing 75 arm curls in a minute. And then not just in a single minute, but doing 75 arm curls all day long. And you don't even have to put a weight in your arm. Just sit there and do elbow flexion and elbow relaxation 75 times a minute, or even 60 times a minute, and see how long your muscle is going to want to keep on doing that within there. Smooth muscle is a little bit different from that of both cardiac muscle and skeletal muscle in that there's no visible striations within them. They basically have very little or no sarcoplasmic reticulum. So once again, their contraction is going to be a lot longer within them. The individual cells have gap junctions that run in between the cells that allow them to contract as a unit most of the time. And so we end up with two kinds of smooth muscle. We have visceral smooth muscle, and then we have multi-unit smooth muscle. Visceral smooth muscle is going to have essentially a motor unit where the neuron comes in and it doesn't actually connect to each individual cell. It kind of goes for the bundling process. And so it's going to attempt to have kind of all of them spread the action potential throughout the entire bundle itself. Versus multi-unit, each individual cell has its own neuromuscular junction. And so every single cell is going to be touched by an individual neuron itself. And so they're all going to have some sort of neuromuscular junction that's there, which means they're going to function a little bit more like a skeletal muscle. And so we find these in areas where we need to have the muscle contract at specific times, and we don't want it to just kind of spread across the cell itself. We want them all to contract at the same time. And so things like erector pili muscles, your iris utilizes them. The arteries, inside our lungs, and so our large airways there where we're going to have bronchoconstriction and bronchodilation, all use a multi-unit type of smooth muscle. When we have smooth muscle in something like an organ, and so especially that of the digestive tract, we'll see that the muscle itself is generally lined up in two sheets. And so we're going to have two different kinds of directions that those fibers are running. And so we're going to have longitudinal fibers where the fibers are kind of all running in this direction. And then we'll have a set of circular muscles that are basically wrapping around the muscle. So if we had a piece of intestine here, we would have some muscle fibers that are running the length of the muscle this way, and then we would have another set of muscle fibers that are wrapping around the muscle. And so they're generating a situation like this. And that's what we see in the picture here, is where we have two types of muscle types that are within the muscle there and generating the contraction in two different directions along the muscle itself. And again, very little sarcoplasmic reticulum, so almost all of the calcium needs to come from outside of the cell. When we look at the individual smooth muscle cells, we see that the smooth muscle doesn't have any visible striations to it. But when we look very closely, we can see that we have intermediate filaments. And those intermediate filaments come together on these little dense bodies. And where these little dense bodies are kind of like the attachments of the intermediate filaments. And so what it amounts to is like having a big net around the muscle. And when the muscle contracts, when that cell contracts, it's going to spin in on itself. And so it's going to have kind of a helical type of a pattern. And the muscle itself is then going to shorten. And so it's going to twist in on itself. The easiest way to think about this is thinking like if you were wringing out a towel and you start to twist and twist and twist that towel, the towel starts to become shorter and it becomes thicker in the middle. And that's what we see here, going from a relaxed state to a contracted state of the muscles themselves that are inside there. And then as the muscle relaxes, it then untwists and the surroundings will tend to then lengthen the muscle again. The contraction itself, since we don't have proteins aligned in the same way, we also have a slight change in the proteins as well. And so within the cells themselves, calcium is going to flow in from outside of the cell. So the calcium starts to come into the cell itself. And when it does so, rather than looking for troponin, it's going to bind to a substance called calmodulin. And so this calmodulin is going to be a catalyst to a chemical reaction that phosphorylates the myosin head, and this allows the contraction to occur. And so we have to do phosphorylation to the myosin head, and calcium binding to that calmodulin initiates that step that's there. But it's a slow process. And so contraction of smooth muscle is relatively slow. We have very long contraction and relaxation times for our smooth muscle. Again, here we can see the difference between longitudinal layers and circular layers in smooth muscle. And so where we see them here, we see a layer of smooth muscle going around this way. And then the outer ones here, the longitudinal layer, are actually in a sense coming out at us. And so those fibers are going to be running along the length of the organ rather than going around it. And what this allows for in smooth muscle for the digestive tract is what we call peristalsis. And so peristalsis is the movement of the digestive tract in by alternating contraction and relaxation. So we can kind of have a process where we contract and relax, and create these waves along the digestive tract that eventually kind of squeezes the contents along the digestive tract itself. And so it's basically squeezing it from beginning to end, kind of like if you were trying to squeeze all the contents out of a plastic bag. Smooth muscle is going to have potential tone in it as well. And so with all of that calcium kind of moving slowly into the cell from the outside of the cell, it does take some extra time for that to occur within that. And so we're going to have a sustained contraction that is going on. And there's also going to be essentially this kind of partial contraction almost all of the time within the muscle itself. And so there's going to be quite a bit of tone there within that. And that's good for doing things like maintaining blood pressure, so that we don't have just again, a on and then off, and on and then off for the muscles. We have kind of this balancing change within the muscle, and we have this overall tone within the either blood vessels or the digestive tract. When that muscle wants to contract, especially within the digestive tract, one of the things that it does respond to is actually stretch. And so as you're eating a meal and your stomach starts to stretch because it's filling and filling and filling, what it'll do is actually cause the muscles of the walls of the digestive tract to relax. And so that's going to make the size of the stomach start to change there. And as the food content is pushing out on the walls of the stomach, that then causes the stomach to increase in its size. And so it relaxes and allows for it to increase in size. As the stomach then empties and it starts to move on to the next part of the small intestines, then there is less pressure on the walls. So there's less pressure there, and the muscles are going to contract, thereby bringing us back down into a smaller state. And so they're going to rebound and cause that area to start to decrease in its size. In other places like blood vessels, we can have local changes especially dictate what's going on, and so we can have chemical changes in our pH level, our carbon dioxide levels, our temperature, ion concentrations can all lead to changes within the contraction of the muscles themselves. When it comes to damage to a muscle cell, essentially we can't fix it. And so the one exception is smooth muscle. And so although we have the ability to kind of regenerate some individual cells here and there, the cardiac muscle and skeletal muscle just does not have the general ability to create healing within a muscle to where we get more muscle cells. And especially remember back for skeletal muscle. Skeletal muscle is the combination of multiple individual cells fusing to one another. And so we don't have that ability. There has been, especially with cardiac muscle, there has been some research that's been looking into the use of stem cells in order to regenerate cells after things like a heart attack. That's there. But for smooth muscle has to be able to go through a process of hyperplasia at times, especially during times of pregnancy when the uterus needs to increase in its size. And so cardiac muscle and skeletal muscle will generally go through a hypertrophy, whereas smooth muscle can both do hypertrophy, increase in the size of the muscle, as well as hyperplasia, increase the number of new cells that are actually there. This is also required when we need to grow new blood vessels. And so as new blood vessels need to grow because we're growing or because we've damaged them in some way, we need to be able to do the same thing in those blood vessels. When it comes to the growth of the muscles themselves, their differentiation does occur relatively early on in life, and we move from the stem cells into growing either skeletal muscle cells or cardiac muscle cells or smooth muscle cells. And this allows for that differentiation to occur. And especially in skeletal muscle, that skeletal muscle continues to grow and add cells and so on throughout the first year or so of life. And then from there, it's essentially use it or lose it. Build that muscle up when we're younger and continue to grow it as that occurs. And then after around the age of 30 or so, we start to see a decline in those muscle fibers if we're not using them. So the actual muscle itself will start to become infiltrated with lipids, with adipose. And so it'll start to fill in the spaces in between the cells themselves. As our nervous system starts to slow down, our muscles decrease in their ability to react as quickly as well. And so our reflexes are going to be a little bit slower. We're not going to have that spring in our step quite so much in a sense as we used to. That's there. And as we age, we also tend to oftentimes, especially in the past, now the trend is starting to change as far as our activity levels goes, but it always was that essentially as we aged, we become slower, we're more sedentary and things like that. And so we're going to start to see a change in the fiber types that are predominating in the cells as well, generally more towards a slow oxidative fiber. But the more you stay active, the less that's going to occur. Some problems that can occur with muscle are things like myasthenia gravis. We talked about this a little bit when it comes to the neuromuscular junction before. And so this is an autoimmune disorder. It tends to affect women more than it does men. They're usually in their reproductive years when diagnosis occurs. And essentially it destroys the acetylcholine receptors. And so the autoimmune starts to block those acetylcholine receptors, which means that even though the muscle is the nervous system is telling the muscle to contract, it's going to not be able to do so because we can't bind that acetylcholine to the receptors themselves. Initial symptoms may be problems with reading, especially for extended periods of time, sometimes swallowing with a longer meal, things like that within the body itself. And then just generalized kind of fatigue of the muscles themselves. And so everyday tasks becomes more and more difficult within that. And this is where anticholinesterase drugs may be helpful with this disease to increase the amount of acetylcholine that's always within the synaptic cleft. Muscular dystrophies, there are a number of different types of muscular dystrophies depending upon the portion that has been affected. This is a genetic disorder, and so it's a gene that alters an X chromosome, which means that males are going to be more predominantly affected than females are, because a male would have to have a baby with a female that has a X chromosome that is affected as well in order for the female to get the disease itself. Depending upon the type, it may strike early on in life, and so by age 5 or so, they may have decreased abilities to use their muscles, start to stumble and things like that. And again, depending upon the type, that disease may progress into essentially paralysis of the muscles, or the lack of ability to use the muscles over time, to where they may be wheelchair bound by their teenage years as well. This does cause an atrophy to the muscle, and so the muscle does start to decrease because of the lack of force that the muscle is able to generate, and so it decreases the number of muscle fibers inside. In addition to having, in a sense, kind of diseases of the muscles, we can also have some things that occur at periodic times to the muscles as well. And so we can have a spasm, and so involuntary contraction of a muscle itself, and so a contraction and then a relaxation. If that contraction stays, we end up with a cramp, and so essentially that spasm that occurred to cause the contraction, and we may see that contraction go through contraction and then relaxation. But if it stays around, we may get a cramp. Tics occur repeatedly, and so this is where the muscle is contracting, relaxing, contracting, relaxing. This is oftentimes seen with muscles that are the facial muscles or those around the eyelid, especially if we're tired. We may see tics start to develop. Tremors, typically a neurological disorder, and so this causes the muscle to contract and relax and contract and relax. But it's not just a single set of muscle, it's typically opposing muscles. So antagonistic muscles of one another are contracting and relaxing involuntarily. If a single motor unit fires off when it's not supposed to, then we get fasciculations. And so fasciculations are a single motor unit. So we're going to see just a part of the muscle actually contract under the skin itself. And usually you'll be able to see just that small portion. It could even occur deeper inside the muscle and you'd never even know it.